So we're now going over to the formal part of the meeting, which is the inaugural lecture for the Richard Peto Professor of Epidemiology, Zhengming Chen, on the 21st century cohort studies, A Long March.
Good afternoon, Pro-Vice Chancellor, friends and colleagues. It's my great honor and privilege to deliver this inaugural lecture on cohort studies. I've chosen this title partly to celebrate the legacy of Richard Orr, who established the principles of larger cohort studies through British doctor studies, and partly also to reflect my personal journey working with Richard Peto, with Rory Collins, in setting up a few larger studies, including China Kadoorie Biobank. I arrived in Oxford 1987, exactly 300 years after the first Chinese scholars who visited Oxford. Okay. His name is called Chen from Nanjing, who spoke very fluent Chinese and spent several months working in boarding library, helping to categorize a larger collection of Chinese books. He was also very popular with James II's court and has his personal portrait commissioned by the king. And for me, I came to Oxford purely incidentally after bumping into Richard and Rory at a tobacco conference in Tianjin, 1987. At the time, I was a postgraduate student working on a small cohort studies. I was really struggling with data analysis because our computer did not have any hard disk. And when I was offered the opportunity to come spend a year analyzing the data, I didn't believe it was real. But finally, the invitation letter came, not by fax, but through telex. And at the time, traveling abroad was a very serious business. So I had to get the formal permission, approval, by the municipal government in Shanghai. So this is actually a red stamp official approval. So it's make me feel I'm a very important person, like VIP. I think the data I brought with me was from a small cohort study called Shanghai Factory Worker. It was established by Professor Li Wanxian back in late 1970s, very much modeled on the Framingham study, trying to study the etiology of cardiovascular disease. It collected a small number of information among 10,000 factory workers in Shanghai, including smoking, simple diet, alcohol, blood pressure, but most importantly with measurements, but no long-term storage of in the blood on the total cholesterol. And when I first get involved, and the study, all the data was still on the paper. So I had to spend a lot of time, get everything computerized by myself, and then subsequently try to establish a record linkage through mortality follow-up, by visiting the factory pension office, by visiting public security bureaus, by knocking at the doors of family members. So this gave me a first taste of running cohort study. To be honest, not very pleasant. But actually, things finally paid off after I came to work with Richard Rory in Oxford. And I managed to get a few good publications, BMJ and JAMA. But most importantly, I started to really appreciate the beauty and power of cohort studies and its relevance not only for China, but also for the UK, and the relevance for clinical medicine as well. So one of the most important findings from my DPhil on the Shanghai cohort study was a demonstration of a positive continuous association of total cholesterol with the risk of ischemic heart disease. And even within the so-called normal range of cholesterol distribution. So as you can see here, within the so-called normal range, you still see a very clear positive association with any evidence of a threshold, suggesting the lower the better. So this actually study finding informed the design of a heart protection study led by Rory and others, and which went to demonstrate the benefits of lowering LDL cholesterol irrespective of the initial findings. So this is a really landmark study. You have heard quite a lot from talks today. And irrespective of your initial levels, if you give 1 tablet simvastatin, it can reduce from high to normal, which reduces risk by 25%. But if someone started with normal, if you reduce, can reduce their cholesterol to below normal, you still, reduce risk very substantially. So this landmark study totally changes the way we view the cholesterol, we treat and manage the cholesterol, but also help to dispel a lot of the confusions which is very prevalent during 1990s about the relevance of a total cholesterol for the health, but also illustrate the importance of a study different population as well. So I think, and also emphasize the need or value of cohort studies. So to this audience, we have heard a lot about the observation study, especially cohort study today. Just to remind us about the strengths of the prospect cohort studies. Survey is done before disease onset, so we can largely avoid or minimize reverse causality. And the probable controls are selected from same population, can largely reduce or minimize the selection bias. If you're able to collect different type of samples and distort them, it can facilitate future discovery research using genomics, proteomic assays as well. And another important aspect is you can study many different disease outcomes simultaneously as well. But for various reasons, especially due to constraints of technology during 20th century, the study done traditionally tend to be small, modest size, with limited data, such as the study I've been involved in Shanghai, and no sample storage because we don't have a freezer at the time. And the follow-up tend to focus only on mortality, not morbidity, and of course, primarily in UK, USA population. But still, they managed to generate a lot of important findings and help to identify many important multiple fiber causes of major disease, such as tobacco, alcohol, chronic infection, blood pressure, blood lipids. So putting together this improved understand disease etiology, prevention, and also new treatment help to improve the health of general population around the world, as shown in these figures. The mortality trend in middle age across 25 biggest countries uniformly start to see very spectacular decrease. But in many countries, this still continue as well, even with the COVID epidemic. But there's still a lot of unanswered questions. For example, causal relevance of a certain risk factors like alcohol, we have heard this morning, and the relevance of particular risk factor in different populations. And for specific disease, sometimes the upwards, downward trend are still not fully understood, suggesting there's still many important kind of causes are still with discovery. For example, this shows the trends in stroke mortality in UK, USA in middle age. Back in 1950s, stroke was very, very common, and both Churchill and Roosevelt suffered strokes. But ever since, the mortality rates start to decrease very spectacularly, nosediving, and at least 8-fold reduction since 1950s. So we still don't really understand what's really going on because blood pressure alone cannot explain this is a very spectacular decrease. But if you go to China, the situation is very different. Each year, 2 million people die from stroke. And also, hemorrhagic stroke is still very common, accounting for 20 to 30% of the total stroke. So in terms of number of deaths, loss of years of life, and of deathly, which is the three kind of measures, stroke is the leading cause in China. So that's still a lot to be learned through large prospective cohort studies. But within China, there are still large unexplained variations in stroke mortality. So this figure showed the student mortality across 65 rural Chinese counties, with a typical population like 500,000 to 1 million. So here you see each letter represents one county. So you have places, 30%, 20% of population will die, at 600 per 100,000. On average, around 10%, by age 70. But you still have areas rates are close to UK. But this cannot be explained by blood pressure, because if you correlate at least the population level, if you correlate blood pressure levels, population level, with the mortality rates, there's no correlation at all. The correlation is 0.06. It's not significant as well. So this is actually quite interesting, suggesting that some important causes await discovery. So this is not only applied to stroke, but many other diseases. Here, esophageal cancer, nasopharynx cancer, you have this hotspot in central China. This has been known for many decades. And back in 1970s, American NIH and NCI put $20 million at the time for a huge nutritional trial. After 10 years, still no clear answers. And the same is true for nasopharynx cancer. They cannot be explained by known risk factors, because this figure shows among women, they don't drink, they don't actually smoke. So suggesting, there's lots to be done to understand. And the study findings will not be only relevant for Chinese population, but also globally as well. So in a way, this reflects the complex etiology of chronic non-communicable disease, nature, nurture, and a complex interplay between the two, which affects the methylation or gene expression, the protein levels, small molecules, physical characteristics, ultimately the risk of developing or dying from particular disease. So to understand this complex disease etiology, to develop new medicines, improve risk prediction, I think the prospect biobank are essential, and for moving from genomes to phenomes and via omic as well. But I think to do that, we need to overcome some of the limitations of a traditional cohort study. The study needs to be much bigger, with a detailed exposure assessment, which are relevant not only for one disease, but relevant for a wide range of different diseases, cardiovascular disease, diabetes, cancer as well. We need to study diverse populations with a wide range of exposures, with different genetic heterogeneities, and with different disease rates. For example, if you want to study stroke, the best place is not UK, but China. Also need to collect and store biological samples, different type samples, blood, urine, fecal, saliva, and those samples collected slightly different ways to facilitate future discovery research. And also able to follow up not only for mortality, but also for morbidity, like hospital record, because this will greatly improve the study power, because you got a lot more numbers, but also enable you to study non-fatal conditions, and also improve the disease phenotype as well, which is essential. So why we need big studies? I think the talk will be incomplete without showing this again and again. This is a slide you see many, many times. I apologize, but I want to make that point again. And so this is the study comparing blood pressure and IHD mortality by age. If you study size only 5,000, which is a unit of Framingham, the picture is very blurred. If you increase sample size by tenfold, become more focused. But if you increase by another tenfold, 500,000 people, you don't need statistics, very crystal clear. But this is only possible by bringing together a study from 61 prospect cohort studies. And it's a huge effort, achievement by Sarah and the team, but this is definitely not the way forward for future. And especially if you want to study genetic other, more modest kind of risk factors, we need a much bigger, much better characterization of disease. So this is standing height GWAS from Kadoorie. If we genotype just 10,000 genotype, we only got 2 hits. If we increase the sample size to 100,000, we got a lot more hits and very definite statistics. If we increase to 1 million, there are even more. So in a way, epidemiology is a number game, but this is critically important as well. So in our welcome development, and many countries start to develop new type of a cohort. So many studies have been established or are being established. But I think this department, led by Richard Rory, are the world lead in establishing big biobank studies. They are big, they have matured, they cover diverse population. So in a way, it reflects the vision of this department, thinking big, thinking long-term, and thinking internationally, and thinking unthinkable as well. So this is summarized a few studies. You have heard Max studies, UK Biobank, Kadoorie Biobank, I'm going to touch briefly, and also part of an EPIC study as well. Plus many ongoing biobank as well, but there will be at least 10 years before you see the main results start to emerge, because you have to be linked to the health record. It's a very slow, long process. So I've been very fortunate to be involved in leading, establishing the Cohort Kadoorie Biobank. I mean, certainly Kadoorie Biobank did not come by chance. It builds on 30 years collaborative research, started by Richard Rory back in 1980s. So here I just list several of the studies, 6 population studies. They're not necessarily cohort studies, some are cross-sectional, some are case control studies, but total over 2 million people involved, and then larger scale randomized trials. Through the process, we have established good collaboration. We understand the local kind of issues and how to establish, run the study. So with the Kadoorie Biobank, it's been not easy process. A lot of challenges. I think many failed attempt in grant application since mid-1990s when I get involved, but actually the first application started back in mid-1980s by Richard and Rory. People thought we were crazy. Why 500,000 people doing 5,000, 10,000? Too big, too risky, and no scientific kind of hypothesis, and also the reference relevance for Western population is questionable, despite the fact that people are quite happy to believe the results from rats, mouse, or zebrafish, but they don't believe the study from elsewhere. But eventually, we managed to approach good friend of department, Judith McCall, who put us in touch with Sir Michael Kadoorie, billionaire, and his foundation. They trust us, and they agreed to fund the study, one-off with a total $7 million at the time. 2 conditions. One is the study needs to bear the name Kadoorie. That's easy. We're all happy to do that. Another thing, try to get the sample, part of the sample, exported to be stored in Oxford. We thought that's achievable, but it's not been a very easy process. It's been a very long, bumpy journey. After more than a year, we're still getting nowhere. The initial application to Chinese Human Genetic Resources Administration was filled 6th of September 2001. And after we approached the Ministry of Science and Technology, they're happy to reconsider. And then Kadoorie Foundation lost their patience. They imposed deadline. You have to get something. Otherwise, the whole support will be withdrawn by the end of May 2002. So in last attempt, Richard wrote to Ministry of Science and Technology, we're coming, we try to discuss, try to get some sort of arrangement. And then internally, subsequently, we heard most of the Ministry of Science and Technology already turned down. They said this is not appropriate, it's too big, too risky. So we have our flight ticket booked on 11th, which is Saturday. On the 10th of May, our colleague in China CDC managed to secure, at our request, a meeting with the Vice Minister, Huang Jiefu. So I was sitting with Richard evening, I said, this is probably very unlikely. We'll probably return empty-handed. But if we got agreement, green lights, there's no time to get, stamps, because the whole process will probably take a week or so. And so I said to Richard, let's actually pretend everything goes well. Let's, get approval letter drafted, so there will be no time delay. And the next day, we went to meeting Ministry of Science Health, and with the charm of Richard, he managed to persuade the Vice Minister. And he said, yes, this looks very reasonable, why not support? So by 5 p.m. in the afternoon, we got this approval letter, red stamped. We rescued the study last minute, and the letter had 175 words. So each word was exactly $40,000. So in a way, in retrospective, to reflect, if you want to achieve something extraordinary, so you have to be very ambitious, very bold, brave, but also very patient, but also with a little bit of luck. I think we struck that somehow. Once we overcome that kind of a hurdle, everything becomes very easy. We managed to set up centers, identify 10 different areas, regional centers across China, from tropical and the south to freezing north, from coast city to inland rural areas, with a very different stage of economic development, different risk exposure, different disease rate. And we managed to recruit within less than 4 years. At the time, Richard said, because recruitment went so well, let's keep going. Let's get 600 or even million. But very soon we run out of the money. But at least we managed to get in 1 month, because instead of 500,000, we managed to recruit 512,000, because at the time, UK Biobank was recruiting, so at least we can actually make sure we are slightly bigger than UK Biobank. So after that, we did 3 resurveys in a random subset 5%, 2008, 2014, and also more recently during the COVID 2021. But actually, although it's a huge achievement, everything done very, very carefully. As Rory said, a big study is not a small study made big. So we have to actually pay great attention to study design, to the detailed procedures and documentation like SOP, but most important, to have a very robust IT system to manage all aspects of study. And also to have regular training, monitoring, reporting to make sure 1,000 miles away, we understand what's really going on. If something happened, we can take action immediately. So this is a book we published a couple of years ago, summarizing what we have learned. If you want to start some new cohort study, I'll be quite happy to give you a free copy after the meeting. And one of the most important aspects is actually the IT system. I cannot emphasize more. So this is actually, we developed over 100 bespoke IT systems covering almost every aspect, not only field survey data collection, but also sample processing, shipment, storage, reformatting. During the whole process, we didn't lose any single samples. Not a single sample gets sold or damaged. And also long-term follow-up, outcome validation, but also other aspects, user center management, remote system management, communication. If the system down, we can set patch overnight and get incorporated, and then will not cause delay with the field work. QC monitoring, asset management, equip calibration. You don't need to remember. System will remind you. So that makes life much easier and a great improve the data quality, efficiency of the whole operation. So just a few sort of screenshots of the system, but there's many of them. It's not possible to go through that one. So this shows the operation, the assessment center with different stations after very, very careful pilot study. So we'll come up with, all the fit out arrangement, the number of staffs, the stations, everything carefully planned after extensive pilot study to make sure there's no bottleneck and the things can move freely. And so overall, each during the assessment, there's 18 different staffs. So to go through the whole process, probably 1 to 1.5 hours, depending, the questions. So overall, we recruit around baseline, a bit more like 65, 70. Second rate survey, a lot more additional enhancement, around 60. So overall, we're recruiting 14,000 per site per year, and 140 sites per year, across 10 different areas as well. So everything actually connected by computer through Bluetooth. So they're synchronized. There'll be no risk of any data loss during the process as well. And then overnight, the data transmit, via Beijing to Oxford for further checking, integration into the system. So the data is very high quality, no missing value, no Roger errors, and you can actually start analyze almost immediately. So after this more than almost 20 years, so the study has become a global resources with a unique combination of scale, breadth, and depth, with a lot of data collected baseline during the 2004 and 2008 questionnaire, physical measurements, environmental data like ambient temperature, and exposure to environment, to air pollution, and most importantly, blood samples. 3 resurveys, repeat everything baseline, but with a lot of enhancements. For example, bone density, CIMT, liver fibrosis, accelerometer, retinal imaging, and a new type of samples as this become possible over the time, urine, saliva, fecal, plus, as well as the blood. And also in recent years, we start to, with technology development, we start to do sample assays, very much like kind of proof of concept pilot study. But ultimately, what we liked is model the UK Biobank, try to do sample assay for all 500,000 cohorts. That would be really powerful. So like blood biochemistry, proteomics, metabolomics, infectious marks, genotyping, we have done 100,000, whole genome sequence, 10,000 as a pilot study. But now the technology is there to do the whole 500,000. The cost is probably £100 per sample. So, but still, 500,000 is still quite a bit of money. So we need to actually, find money to do that. But most important things for the success or failure of a cohort study is follow-up for disease outcomes. In the Kadoorie, we are quite fortunate to be able to link not only to cohort-specific mortality, but also to disease incidence and not to hospital record, like health insurance. So now there are over 70,000 deaths, all ICD-10 coded, and with over 1.5 million episodes of hospitalization, but only 1% lost to follow-up. This is a demonstration of a careful planning and also excellent work by a colleague in China as well. So this shows a number of selected incident disease. Some are very common, like ischemic stroke, as mentioned. We have recorded almost 75,000 incident cases of stroke and 16,000 hemorrhagic stroke. 93% of them confirmed by CTMI, so it's very definite. So with that, we start to see a lot of interesting findings, construct patterns of association that emerge. I will come back to that one. But also with many other diseases in, cancer, 40,000, diabetes, and some actually many non-fatal disease. They are not extensively researched because it's just not possible to link to them. Arthritis, cataract, and asthma, osteoporosis, retinopathy. And as population is aging, we also start to get a lot of neurodegenerative disease like dementia, Parkinson's disease, and even snake bites and earthquake that struck one area in 2008. So this demonstrates the quality of linkage and the completeness of our record linkage as well. So we don't really stop there. For major disease, we also try to go back to retrieve origin medical record and to try to improve the disease phenotyping. And at least for 4 major categories of disease, stroke, IGD, cancer, and chronic renal disease, we have retrieved over 100,000. This greatly improves the disease phenotyping as well. And this is a huge resources. After a short period of exclusive use by us, colleagues in China, we also made the data widely available to the global research community. So all the baseline, first, second rate survey, follow-up data are now available. You can actually request. So Internally, we also do a lot of research, hypothesis testing, discovery research, and with many interesting novel findings as well. So in total, we have published 450 papers, still a long way to catch up with UK Biobank, but I think with a lot of interesting novel results. So in the interest of time, I'm just going to focus on one particular disease, stroke. So this shows risk exposure. We have analyzed, published, lifestyle factors. Some are very obvious, well researched, and also biochemistry, like blood pressure, LDL, HDL, metabolomics, and genetic, so GWAS, but also using single genetic variants to inform the causal reference and also drug discovery, risk prediction, and also long-term prognosis and healthcare utilization. So actually, it's very a broad and research kind of area as well. And with many expected and some unexpected findings. So for some, we just confirm what we already know, but the few evidence get for China. For some, results are much more certain compared with the previous data, especially for stroke. Our data probably more than double the worldwide evidence in some cases. But for some, they challenge the convention, really novel. So I'm just going to through very quickly in a few examples highlighted in bold. So the first one is a stroke kind of prognosis. Very poor prognosis. Although the incidence rate is very high, mortality is high, even you survive from hospitals, discharged alive. So within 5 years, 41% will have another recurrent stroke. 70% would die, and which is particularly high for ICH, hemorrhagic stroke. So this is illustrate the poor management risk factors like blood pressure and also inadequate use of proven second prevention as well in China. So this paper was published a couple of years ago, Lancet Global Health, with invited commentary, highlight importance of pay attention to stroke because there's millions of people with stroke survivors as well. So we have heard from Robert this morning the importance of blood pressure. Again, we see very clear, strong positive association, and no evidence of any threshold. The low, the better. And the association appeared to be twice as deeper for hemorrhagic stroke than for ischemic stroke. So this is association is definitely causal, as reported, demonstrated by Robert's talk, which is published early this year with editorial as well. And then come to adiposity. It's very interesting. High adiposity, high blood pressure, high stroke risk. So when we analyze association of adiposity with stroke, for ischemic stroke, this is what we expect. This is without adjustment for blood pressure. This is with adjustment for blood pressure, suggesting that the effects mainly operate through high blood pressure. So we would expect very similar patterns for hemorrhagic stroke, but instead what we see is very strange patterns. This is not analytical error. We checked, and the check is definitely real. So for hemorrhagic stroke, there's little association, between below 25 and to 17, where the blood pressure differs by 12 millimeter mercury. This would correspond to 65% high stroke, hemorrhagic stroke risk, but it's complete flat. If you adjust for blood pressure becomes strongly inverse. So this is suggesting there's something going on, offset the adverse effects of high BMI on high blood pressure. We don't really know what it is. So this probably answer will come from sample assays, metabolomics, proteomics, chronic infection as well. We also demonstrate the benefits of certain lifestyle factors, fresh fruit, which is published in the Journal of Medicine, and also physical activity published in JAMA Cardiology with invited commentary. Of course, it's difficult to establish causal effect association, but at least this is a lifestyle factor that have many other beneficial effects as well. We should encourage people to eat more fresh fruit and to do more exercise as well. Another thing I'd like to mention is the old story, plasma homocysteine, which is the amino acids required for DNA protein synthesis, has been associated with increased risk of cardiovascular disease during 1990s, 2000, and subsequently several large randomized trials, including the SEARCH study, and tried to test the supplementation of folate, which can reduce the homocysteine. But all this trial found no evidence of any benefit by supplementing. But it's mainly in European population where the population has a much higher level of folate because of diet or because of a mandatory folate fortification in the flour. But in China, we don't really know its relevance in China for disease risk, because in China, the average folate level is only 1/3 of that seen in UK, US population. So to answer that question, we can use a genetic like MTHFR polymorphism, which increase the homocysteine by 20 to 25%, especially if the folate level is low. And now we did a Mendelian randomization, used a very simple genetic variants, and on stroke and other disease. So this is the study findings, and the paper is led by Derek, Robert, which just sent back to IGE, hopefully they will accept very soon. So for ischemic heart disease, complete flat, consistent with the randomized trials, but for stroke, very clear excess risk if suggesting it's likely to be causal. So 23% increase for TT versus CC, and although ischemic stroke, 11% is very highly statistically significant. For other disease, no any excess or reduced risk as well. So as a result, we've been in touch with a group in China, stroke doctors. They're now in the process of setting up a new folate stroke prevention trial. So folate supplementation could be used not only for prevention neurotube among newborn babies, but could also be considered as a primary stroke prevention in China, other countries as well. I'm now going to turn to alcohol. You have heard from this morning's talk. So with alcohol, we all enjoy a drink after a busy day or big celebration like this. But one big question has puzzled epidemiology the last 50 years is whether moderate drinking is beneficial for heart disease, stroke. I think a randomized trial was planned but got cancelled in USA a few years ago. So to answer that, we can bring in genetic epidemiology or Mendelian randomization. So this is the findings from genetic studies of alcohol drinking and stroke among Chinese men, because Chinese women, they don't drink. So what we found, there's any evidence of a so-called J shape or U shape. So this is moderate drinking, 1 to 2 glasses of wine, which increased stroke risk by 12%. Very definite. There's any evidence of a so-called protective association. So the key finding from genetic analysis is using very powerful genetic variants present only in Chinese East Asia. We have demonstrated more than 64 difference in alcohol intake and across 6 main genetic categories, but no difference in other potential confounding factors. So like education, smoking rate. So 1 to 2 drinks per day increased stroke risk by 12%, higher for hemorrhagic stroke than for ischemic stroke. So overall, among Chinese men, 16% of stroke could be attributed to alcohol. And this association among men is not due to pretropic effect of a genotype, because among Chinese women, they carry same genotype, but they don't drink. We found any evidence of association with a stroke, with the blood pressure, with HDLC as well. So this is actually really nice. And when we look at conventional epidemiology, again, this is a familiar patterns and subject to all kind of a bias confounding, even with extensive effort to try to control. So typical J shape or U shape for ischemic stroke, and for hemorrhagic stroke again. But in genetic epidemiology, when we manage to control all the potential bias, you see a log linear positive clear-cut evidence with no evidence of any safe drinking unit. So the paper was published Lancet a few years ago with editorial, and this morning you have heard our more recent work, Nature Medicine, and it's indicating the risk is beyond stroke, but also many other diseases. So I think this is really important, especially for tonight. Don't indulge yourself. You can propose a toast for friendship, for prosperity, for papers, but not for health. And then finally, a few slides on well-researched biomarker. LDL cholesterol, you have heard, and also related to my first cohort studies. For ischemic heart disease, yes, no controversy. But for stroke, the epidemiological evidence has not been clear-cut, partly because old study cannot differentiate clearly different type of stroke. So once you're able to do that, we see very different patterns and almost mirror imaging for ischemic stroke, clear positive, even within the so-called normal range. This is below 3.5. You still see very clear positive association with no evidence of threshold. For hemorrhagic stroke, complete opposite. But the question is this causal? And the answer is yes, because we also bring together genetic, we also bring together all the randomized trial evidence. They're highly consistent with each other, suggesting this real or causal. And the paper was published a few years ago, Nature Medicine. But overall, despite the adverse effects of cholesterol lowering on hemorrhagic stroke, overall, in China, there's still big net effects or benefits as the LDL's cholesterol lowering as well. So this is important. We guide you how best to use that effectively, safely as well. When we look at HDL cholesterol, in the previous study, prospect study by Sarah, demonstrate the inverse association with ischemic heart disease. And in Kadoorie biobank, we demonstrate very clear inverse association with ischemic stroke, with hemorrhagic stroke complete flat. But again, the question is this causal? And the answer is no. So we use the genetic epidemiology, particularly CETP variants, cholesterol ester transportation protein, which we have identified in a couple of functional variants, which if you carry variants, your HDL level substantially increased, very definite, but has no effects on the vascular disease risk. So CETP defect versus not, relative risk based on 25,000 incident cases of cardiovascular disease, stroke, IHD, complete flat. If anything, suggests that could be some kind of excess risk associated with eye disease. So the paper was published, JAMA Cardiology. Results are highly consistent with the trial, including a trial done by this department, showing no net benefit of HDL-C raising, but does not cost $1 billion as well. But this is actually two biomarkers which is subject to extensive research over the last 50 years. This is not the way forward, trying to accelerate. And with the advent of a technology, now we're able not only to measure these few biomarkers, we can measure a few thousand or even 10,000 proteomics, metabolomics. So this is actually our last few slides I'm going to share with you. It's a proteomic pilot project we have recently completed. So protein is essential for the tissues, the structures, regulations, and the function of the tissue and the organ as well. So in the blood, there's a various type of protein circulating. Some are secreted by blood cell, some are actually actively secreted by tissue, some are actually due to damaged tissue or a membrane as well. So you can actually detect large number of proteins. This is a technology we've been monitoring, following for the last 10 years. And we managed to get department support through Rory and some other small funding and to undertake this pilot study involved 4,000 individuals and all genotype with no statin use, 2,000 IHD cases, 2,000 subcohort controls. We managed to quantify 10,000 proteins, 3,000 through Olink platform, and 7,000 through SimiLogic, covering transport proteins, antibodies, enzymes. And this is actually so exciting. This is something we've been waiting when we planned the study 20 years ago. And there's so much going on, even with the small sample size. We're doing genetic GWAS, understand the genetic architecture proteins, drug target discovery for different disease, IHD, diabetes, adiposity, risk prediction, early diagnosis, and also mechanism linked to known risk factor, like you have heard from Auna this morning on the alcohol effects. and also help to clarify molecular pathways of known genetic variants. Although you have identified hundreds of genetic polymorphisms for IHD, but most of them we don't really know how they operate, what's the mechanism. So this protein can help clarify. I'm not possible to go through all this. We have a special research symposium planned. It will take place Friday this week on human proteins and health. So it's already oversubscribed. But if you're desperate, want to participate, let me know, or I may be able to squeeze you in. So just one final slide on the proteins. So this is the O-link, 2 batches, each with the 14,000 proteins. So this shows the associations with the risk of IHD. And this is after adjustment for all the conventional risk factors, after adjusting for multiple testing. So anything below this dotted line suggests they are significant. Red means positive, and blue means inverse association. So we have a lot of proteins. Each dot represents 1 protein. I can only name a few. And unfortunately, PCSK9 got buried, and somewhere in the middle because the department is doing large randomized trials on that. So this is actually very, very exciting. And for SomaScan, we have more than 800 proteins significantly associated with that. Of course, this is observational analysis. We need to establish the causal effect. So this is illustrate the analytic approaches for discovering novel proteins biomarker. So this is kind of a new type of epidemiology. No longer just observation. We're trying to understand, is this causal? Is this real? Is this relevant for drug target? What's the mechanism involved? So this is illustrate the analytic approaches we have taken for IHD, genetic instruments through GWAS, 2-sample MR analysis, tested in global consortium. Causal proteins were identified 13 UKB, CKB, 16 UKB, and with probably 4 or 5 overlapping, so they're very real, the potential novel drug target. And also try and undertake various downstream analyses, PHIWAS, knock-down mouse, oncology, ontology, functional GWAS, drug target, try really narrow it down. So this can be put into clinical translation rapidly without waiting for another 5, 10, 20 years. So this is just beginning, and I think it's really, really exciting. So in a way, we are in a very, very exciting era in terms of biomedical research. I think epidemiology at the forefront. And it's never been a better time to study epidemiology. And there's no better place than Oxford to study epidemiology because all these large biobank studies, they have matured. So together, they will transform our understanding of disease etiology. We have many, many discoveries about the new medicine, new ways of treating, preventing, and new ways to diagnose and also predict the risk. But the key things, there's still a big questions, how to get the sample assayed. It's a big challenge. But technology is there. We can get whole genome sequence done within 2 years at a cost of 50 million for $500 million. Proteomics is coming. So if anyone knows someone with a deep pocket, let me know. So this is really, really exciting. And I'm going to stop here, finally, to thank all the colleagues, collaborators, founders, particularly my mentor, Richard and Rory, for inviting me to come over to Oxford last century, 1987, for enabling me to stay, and for mentorship, inspiration, and also encouragement. And finally, I'd like to thank my family, my daughter, who came unprepared, unplanned when both my wife were doing PhD, but she has really grown up, gave a lot of joy, happiness, but also a lot of early days, sleepless night as well. She has now qualified in medicine, just got married last month, and hopefully my status will be upgraded very soon. And my little boy, and now a young man, he's also in the audience with his fiancée. And finally, most importantly, to my wife, Yiping, who is also a colleague, for the love and for the sacrifice, for the support, and for spoiling me with delicious Chinese food. This is something I want to repay back over the next 10, 15 years. And also, finally, to my parents, my late father, who died sadly a few years ago, and my mother, who survived the COVID, and at age 93, but still a good health, and for giving me the life and for raising me up during extraordinary, difficult period of time in recent Chinese history. With that, I'm going to stop. Thanks very much for your attention.